So this is one of the modified Mendel monohybrid ratio. Here, like incomplete dominance, codominance is also a modified Mendel monohybrid ratio. That let me remind you once again the Mendel, what I said, whenever the two allelic forms of a character are crossed each other, in case of Mendel, we say, whenever the two allelic forms of a character are crossed each other, only one of the form will come for expression. Okay, so here, let us see what happens in case of codominance, then how it differs from the Mendel. We'll go for. Now here, we are taking the example for this particular factor is blood groups. Let us see the example first. Then after we'll discuss about this codominance. How it is codominance that we have to see. A little brief is required over about a sugar polymer. We have to discuss that above on the RBC, above on the RBC, a sugar polymer that called A factor, B factor. Likewise, that two types of factors are present. Based on that, we can classify the RBC of human being like this. Let me show you. Blood group inheritance we'll discuss in detail afterward. But we are talking about a sugar polymer which is present in the cell membrane of RBC. Some of them are having factor A as a sugar polymer. Some of them are having factor B. Some of them are having factor A and some will have the factor B. Okay, factor A in some and in factor B in some. Okay, then some of them will have both A and B. How it is that we have to see now. A and B. Okay, then some don't have any kind of sugar polymer on the surface of RBC. It doesn't affect any oxygen transport. We'll discuss blood groups later. So with these, just for our necessity that I am going a little information about, this kind of blood group is called A blood group. Those are having B factor, B blood group. And those are having both factors, we call them as AB blood group. If there is no any sugar polymer that above on the cell membrane, Dominant's membrane of RBC, we say that is O factor. That is O. Okay. So these are the four blood groups. In that, I am taking the A blood group and B blood group. Imagine A blood group and B blood group, A and B are crossed each other. A blood group person is crossed with the B blood group person. A blood group person is crossed with B blood group person. Let me take here that A gamete here, B is a gamete. Okay, so that when A and B are crossed each other in the F1 generation, which one is expressed? A expressed or B expressed? Both are expressed. Let us see their RBCs. There you can understand how both can come for expression. This is a person, father. This is mother. Whatever, mother or father, no matter. Doesn't make any difference. As I said to you, there is a factor called A that on the surface of RBC, factor A present. Here on the surface of RBC, the factor B present. Okay, people call this as antigen. Why it is called antigen? That all we'll discuss in the blood group inheritance. So that A sugar polymer containing father, B sugar polymer containing mother, that when they cross each other. So what kind of RBC is obtained here? That we have to see what kind of RBC is obtained here in the F1 generation is containing both factors. That A factor is there, A factor is expressed, and also the B factor. B factor is also expressed. That means AB blood group we use it to say. So which one is expressed whenever the two allelic forms of a character cross each other in the F1 generation, both are expressed. Codominant factor, we say. It has codominant factor. So we continue the experiment here to understand it much better. When F1 crossed with F1, that means AB crossed with AB. So gametes will take as usual A and B. Here also A and B. So I am taking the gametes here into the boxes A, B, A and B. Okay, so let us write. Let us write that what will
Capital,
we have to write. So Japani, first one, capital
capital A, capital
means A blood group.
Capital A, small capital B.
That
AB
means
AB
blood group.
AB blood group. Okay, next here. AB
B
blood
AB
cap
blood group.
B blood
here.
group. BB blood group.
B blood group. B. We can say expression as B. Okay, now the same again. Phenotypic ratio. Phenotypic ratio and genotypic ratio. We have to see the both here. That phenotypic ratio and genotypic ratio. Okay, phenotypic ratio. So, how many A blood group appeared here?
Three.
One.
One.
How many AB blood group are appeared?
Oh, two.
Two. How many B blood group are appeared?
One.
In the genotypic format also you can see homozygous A, heterozygous one AB, and homozygous one B. That means phenotypic ratio and
One
genotypic
is to two.
ratio both are going to be same. Like incomplete dominance here also, we can say both phenotypic and genotypic ratio are same. And this is also considered as modifications of monohybrid cross. Modifications of monohybrid cross of Mendel, which is not following that Mendel law of dominance. That means according to the law of dominance, the two are crossed each other, only one should come for in the F1 generation, isn't it? But both came for expression. Both came for expression that in the codominance, both came for expression. A very interesting thing here that when we cross A blood group with O blood group, it follows Mendel. That means we get AO blood group. This is law of dominance. When A blood group persons are crossed with O blood group, in the F1 generation, we got A blood group. That means no expression of O, only A gene is expressed. So A blood group will come. When we do the cross F1 with F1, AO crossed with AO, that means heterozygous A crossed with heterozygous A, then in F2 generation, that AO, AO, AA, AO, AO, O. That means in phenotypic ratio, 3 is to 1 are appeared. That means 3 A blood group person, 1 O blood group person. Genotypic ratio, 1 is to 2 is to 1. Okay, 1 is to 2 is to 1. So that is the interesting thing here. In the same way, when we do the cross between the B blood group with O blood group, B, homozygous B, crossed with O, what we get in the F1 generation? B blood group. Okay, B blood group will get, when we cross the BO with BO, then in phenotypic ratio 3 is to 1, genotypic ratio 1 is to 2 is to 1 will come. So that means in these two cases, Mendel law of dominance is applicable here. In the two cases, that A cross with O, B cross with O, Mendel law of dominance is applicable. But interestingly, that in this particular area, the codominant factor, both are expressed over. So, if we ask a question is asked over what blood group inheritance is, whether it follows Mendel or not, that blood group inheritance. Okay, we'll discuss the detail in blood group inheritance also, but very interesting thing and asked in the previous question, what it follows, blood group inheritance follows. Blood group inheritance follows both blood group inheritance follows both law of dominance of Mendel, in which cases that when A blood group crossed with O and B blood group crossed with O, it follows Mendel. 100% A come, 100% B will come. Okay, but A and B are crossed each other. What will get? AB will get. That means this is law of dominance and comes codominance. So that is the interesting thing here. Law of dominance is there, and also blood group contains codominant factor is there. Codominance is there, law of dominance is there. And one more interesting thing, till the time we are seeing Mendel only. That means when we are taken the seed color, we say yellow, green, two allelic forms. Okay, when we are taken seed shape, round and wrinkle, two allelic forms. Then when we are taken the height of the plant, tall and dwarf, only two alleles we are taken till the time, isn't it? So now when we come to this particular area, when we come, in case of blood group inheritance, as we are seeing, three allelic forms are present. You see here, there are three allelic forms, A, B, and O. Three allelic forms are present. A form, B form, O form. Not like Mendel. It is called multiple allele. So we should say it as multiple allele. Okay, so this is called multiple allele. So blood group inheritance characters we can say like this. That having three allelic forms for a character called blood group. That's why we say multiple allele. In some formats, it is expressing law of dominance of Mendel. In some formats, it is expressing codominant, which is against the Mendel. All these are the properties. Okay. So this is codominant. Here we just focusing on codominant, but little interesting thing. That's why we are saying that this is about the codominant. That phenotypic ratio 1 is to 2 is to 1 and genotypic ratio 1 is to 2 is to 1. Okay. So this is about the codominant